assignment, we're going to deal with some of the methods used to study transcription factors. And these are really methods that some of them are used to understand the transcription factors. And your book covers most of these in Chapter 7, so we're dealing with Chapter 7 material. And also, there are a couple that aren't covered by it, but these are the ones we're going to focus on. And I've organized them in a way where we could say this is, some of the steps a scientist would take to discover, what is the transcription factor that is binding to this gene we're studying. So we're studying a gene we know about. Let's say it's myogenin. We're like, what transcription factors bind to it? So if we didn't know what MyoD was, we could kind of use these methods in a series of steps to discover those. And so we're going to learn that. And here's what we want to do in this assignment is for the homework is to answer these questions as if they would be on an exam. So we're trying to learn this material in preparation for exam. So that's in part what the purpose of this is to learn to answer these. And each of these would be a 2-point question like that on an exam, like that, okay? So pretty brief, but what would you pay attention to? The highlights. What is the key feature here? What's being done? What's the kind of the pivotal step in the method or the tool that's being used here? And so we're going to go through these one at a time. So the first one is DNA deletion studies. And what we're going to do is we're going to pretend we don't know about the estrogen receptor. And so the estrogen receptor activates many target genes at the time that female vertebrates start sexual maturation, or puberty in humans, right? So a bunch of genes get turned on. So that's what we're studying these genes. And the gene we're going to pick on is actually a little bit surprising because it's not in us or mammals generally, and it's called vitellogenin, and it's referred to as yolk protein. So this is a protein that's expressed, and the example we'll think of is in chickens and other birds, so vertebrates like us, and the estrogen receptor binds to it and activates it when the chicken is going to be sexually mature and is going to start making eggs. And so there's a lot of protein in eggs, and that's a lot of it is this yolk protein, and it's simply a way to put a lot of amino acids into the egg, because inside that egg shell, that hard egg shell of a chicken egg, we got to make a chicken, a little baby chick, and it's got to hatch and on its own power and get up and stand up, and walk around and find some food. So we got to make a lot of proteins, and this is how it's done. The female mother, produces a lot of this yolk protein. It goes into the eggs. Okay, so one of the reasons that's interesting is that mammals don't have yolk protein because we have tiny eggs, right? We have a womb. Mammals have wombs, and as a result, we don't have to put that nourishment into the egg. So there's a term we're just going to learn, and it's called a pseudogene. And pseudogenes are non-functional remnants of genes. So mammals' ancestors were things like reptiles and so forth, and they had eggs and they had vitellogenin, and that gene is slowly being lost. And in mammals it's actually pretty hard to find, but there were mutations that just randomly accumulated in it, and initially it was damaged because it wasn't being used, and it was further damaged, and it just takes a long time, millions of years, for it to really disappear. And it's almost disappeared in mammals, and so just take a look at this, because there's interesting deals about this. There are egg-laying mammals like duck-billed platypus and spiny anteater, and they actually keep that yolk protein vitellogenin gene going. So the gene we're going to talk about is vitellogenin, just because it's not only about humans, right? And actually has some relevance to humans through these pseudogenes. And this protein that binds to it, we know it's the estrogen receptor, but what if we didn't? Can we find it? Can we discover it? And so here's just a little statement about pseudogenes like that. And so, just touching base on those in this example. And so this is what we know the gene is going to look like. So the vitellogenin gene is going to have a transcription unit, going to have a nearby promoter, and this distal, further away enhancer. And we don't know exactly where it is, but it has to be attached to the gene. And so the first method is called DNA deletion studies, and we're going to remove parts of the cis regulatory region. I just want to point this out that historically people call these promoter deletion studies, and this is an example where not everyone has always used the best terminology for the two parts. So we got a transcription unit, promoter, enhancer. This is the cis regulatory region. That's the best name for it. But it used to be that some people called this whole thing the promoter, and that's where this came from, is we're studying the promoter. And they would call this the proximal promoter, that means nearby, and this, I just used the term, the distal promoter, like that, okay? And not many people use that today like that, but these are often called promoter deletion studies, and we shouldn't let that confuse us. And I mention that also because we use these very crisp and defined terms in our course, but I've mentioned before, in history and even currently, not everybody uses these terms. The story's the same, and you just need to be sensitive to that reality. So we might call them promotion deletion studies, and we're going to call them cis regulatory region deletion studies. Because what we're going to do is we're going to, in a test tube, here's our vitellogenin gene. That's what we're looking at, and we're just going to make examples where we chew up. We use an enzyme called a nuclease, and we're going to chew up this DNA from one end toward the other. And so we're going to chew off, in our first example, a little piece, and then a little bit more. And that's what we mean by deletion studies. And then a little bit more, like that. See, like that, on down the line. Okay? And what we do is we chew this up with this nuclease. Nucleases can attack DNA, and this is a DNA nuclease, or RNA nucleases. And some attack from the ends, and if they do, they're called an exonuclease. So we're going to use an exonuclease. And in this video, we're also going to see an endonuclease. So one that attacks randomly from the middle. It cuts the DNA like a scissors, and this one cuts the end like it's chewing it up. So we just add this enzyme for different lengths of time in a test tube, and we don't know exactly what we got, but we got shorter pieces, and then we put these in to a cell and ask it if we can still get the vitellogenin gene induced. And we're really going to be mapping this, the location of this enhancer, because in this first one, still have this enhancer. We don't know where it is yet, but you'll see we're going to map it right here, because the next one also have it, just barely, and then this next one, we would not get induction of the vitellogenin gene in response to adding what? Adding estrogen, right? You can't get it turned on. We've lost that estrogen induction, and so we just say the enhancer we're interested in is in between these two deletions, right? So we've mapped the enhancer of what makes this vitellogenin gene responsive to estrogen. Great, that's what we're interested in, and now we have a piece of that DNA, okay? So that's actually the first example, is this DNA deletion studies. We're mapping using deletions where the enhancer is. Okay, then here's the next one we're going to study, and it's called nuclease footprinting, and we're going to use a DNA nuclease, and sometimes people abbreviate that. They'll say it kind of cute. They'll say it's a DNA nuclease, and this one is going to be an endonuclease. And so what we do here in a nuclease footprinting assay is in our cells, we're going to break them up, and we're going to get the nucleus. So we're going to prepare a nuclear preparation of it, so like we talked about in chapter 1. And then in a test tube, what we do is we have good intact nuclei, and for short lengths of time we can add this nuclease to those these nuclei, this nuclear prep. And it can go in, and it will start biting, right? It'll start biting the DNA, right? And we just do it for a short time, and we get these random cuts, right? And the idea here is that this protein we're interested in, we're trying to discover the estrogen receptor, it's going to be bound here to the enhancer, and we already have a feeling where the answer is, but this is a different kind of experiment. And what we'll see is we should get lots of cuts here and here and here, but this should be protected. And people will say that the estrogen receptor is making a footprint. It's protecting these from cuts. And so what we do is we label the DNA at one end, and we do the experiment where we cut it randomly just for a little bit, get some cuts in it, and then we run that by gel electrophoresis. And this is our control experiment where we did not have any, well, it's hard to describe the control experiment in this example using estrogen receptor, but we have a control experiment just to show, I guess this would just be the DNA, pure DNA, right? Pure DNA in a test tube, and we add the nuclease, and we should get random pieces like that. And they're labeled at one end, so it just shows the longer pieces and the shorter pieces and the shorter pieces like that, okay? And then this is the one where the DNA is studied when it's in the nuclei, and it has that unknown transcription factor, and this is called the footprint. And knowing the length of this fragment, we can map where the footprint is. And if I scroll back to this, we find that that we get a footprint right here. So independent evidence that there's something going on here, right? This is where the inducible faction is, but we can also see something bound to it. But with the nuclease footprinting, it prepares us to move on, right? Because we say, yeah, so we got evidence that that's where the estrogen responsive function is, and that's where there's a protein binding. We know there's a protein binding. So next experiment is this gel mobility shift assay. And so in this, we also prepare the nuclear extract prep, and then we purify the nuclei, and then, oh, I have to move forward. Am I missing this? No. And so then what we do is we take the nuclei and break them open. And now we have what we call, the first thing is called nuclear prep. We prepared some nuclei. And the next thing is called a nuclear extract. And from our nuclease footprinting, we got evidence that there's a protein in the nucleus that can bind to this part of the DNA, right? So now we started with a gene, and now we're really focused on this region that we will find the enhancer in, because this is where the footprinting was. And what we do with the gel mobility assay is all in a test tube, is we just take just this short piece of DNA, like that, and we take this nuclear extract, and we can add the extract to the DNA or not. So those are our two experimental samples, just showing this, and we simply mix it in, and then we do a gel electrophoresis. So it's a gel mobility shift, and the very simple concept is now we have this piece of DNA we're studying, just a very short piece of DNA, and it would go through a gel. The gel separates it based on its size, and it would move through the gel pretty quickly. And when we add the nuclear extract, if we can get conditions where the transcription factor, again, it's the estrogen receptor, binds, it will be a bigger molecule and it will move slower. So this is a nice natural naming of this method. It's a gel mobility shift. So we're going to shift the mobility, and this is what the result is like, is without the protein, we get the DNA moving through the gel pretty quickly, and with the protein in that nuclear extract, it moves slower. What's cool about this is now with the gel mobility shift assay, we've got more evidence. There's a protein binding to this piece, and now we see that it's binding even without the nucleus, right? We can break it open, and we have the proteins just in solution. That means we could do the next step, and the next step is affinity purification. And what we're going to do is we're going to make a tube with some beads in it. I'll just show you what that looks like. So it takes a tube with beads in it, and on these little tiny beads, you can think of them like little grains of sand, like that, and on the beads, we've attached this piece of DNA. And then we add the mixture of that nuclear extract. And the tube has a little mesh at the bottom, so the beads can't go through, but the liquid can. And we start letting them through, and everybody goes through except that protein that we know binds that DNA from the gel mobility shift assay. And we know there's a protein there in intact nucleus. There's something that binds there, and we know from DNA deletion studies that's the piece of DNA that makes this gene responsive to estrogen. So that's what we're after. We're after that receptor that is a transcription factor, like that, okay? All right, awesome. And so what we're doing here is we're purifying this new transcription factor because everything else flows through, and this is called a column, because it looks like the column in the front of a big bank like that, or a big old building, right? Like that, okay? And this is called affinity purification. And it's a type of column chromatography, so it's a type of chromatography. And you've learned quite a bit about that in USM chemistry classes. And this is a type of chromatography that's called column chromatography, and it's a type of column chromatography that's called affinity. We're going to purify things based on their affinity, and the protein has affinity. And look, here's what comes off, and the example in your book is a transcription factor that was studied by Bob Tejan, and there's a box about Bob in the book in chapter 7, studying SP1. We're not going to talk about that. This is purified estrogen receptor. And we probably would start calling it an estrogen receptor because, it causes this estrogen response of the vitellogenin gene expression. So we purified this, okay? And so now we've purified it, and the next thing is we could study it, but now, well, what is the protein, and so there are ways of sequencing the amino acids that are protein and identifying the gene, and we won't look at those today. What we're looking at today is, let's see if we can study this protein by taking the purified protein and actually injecting it into a host animal. And typically that in science they use a mouse or a rabbit. They inject it in, and the animal will respond to this like it's a foreign molecule, and it'll make an immune response. And the immune response is going to be that it's going to make antibodies. And after a month or so, we can have that animal donate blood, just like we donate blood. And so donate a little blood and purify these antibodies, and these antibodies can be used to study the protein, because these antibodies will bind to the protein and help us identify the protein. So this last method is called CHIP, and CHIP stands for chromatin immunoprecipitation assay. And so what we're going to do here is this. In this case, we're going to have some beads, and we are going to have this. In this case, the antibody is going to be on the beads. So the antibody from, we'll say, a rabbit, and we're going to make a column like that, there's another way of doing this that we'll mention here too, and we'll get the protein to stick. And instead of purifying the protein, you'll see what we're purifying is the DNA it sticks to. So let me explain that. So this goes through this story. So we have to first have our antibody, and people will draw antibodies looking like this because they are made of multiple proteins, chains, or they often will draw it like this because this is the place where the antibody binds to, in our case, this transcription factor protein we're trying to study. So we have this. And so the steps in this are just take an intact cell and add a cross-linking agent. And the commonly used cross-linking agent in CHIP is formaldehyde. And that's a preservative, and it works by making covalent cross-links. So if we go back to here is our original example of the gene. So here's this, and we have the estrogen receptor, the transcription factor bound. And what we want to do is we want to make sure it stays bound, and the formaldehyde makes covalent cross-links randomly, quickly, between proteins and DNA, or proteins and proteins, or DNA and DNA. And so we have to use the formaldehyde for just a short time, and it makes these covalent cross-links. And that's good because then, after we do that, we can break the cell, we can isolate nuclei, then we can break the nuclei open, and we can even use a mild detergent to make sure that all the proteins are released from the DNA except ones that are covalently bound. And then we can add our antibody, and we can collect that antibody complex. And in this case, if we ran a column, like we did before, in this case, what we have is we're going to be collecting on the column that protein again, but in this case, the protein will also have DNA bound to it. So what we are doing is we want to study this here, okay? And we want to study the DNA. So then we're going to release those from the beads, and we'll have purified protein with the DNA. Then we can reverse the cross-links, so we can let it go, and we can get rid of the proteins. And now we have DNA. And what we have here is a bunch of DNA, and it's drawn up here. We have all the target genes. And that's what the CHIP assay is used. It's used to identify all the target genes of a transcription factor. So one of the target genes would be vitellogenin, if we were studying it in a chicken, in a hen, like that, and we can read the DNA letters of this and identify all the target genes, So CHIP is used to identifying all the target genes. And so that's a way to further study this transcription factor and what is it doing. And so it, in the case of us and chickens and so forth, there are lots of target genes, and we've talked about that. We've talked before about how one transcription factor can be made. In this case, it's the estrogen receptor, and it activates many target genes. And it can do that because there are many molecules of the protein that get made in the cell when the transcription factor is present. And so there's back to that, so that's the end of this. And so loop back and answer the questions at the beginning and email those to me, okay? And that's the end of this assignment, all right? Great. The end.